Okay, thank you, and thank you to organizers who have put together this very fantastic meeting. Okay, so even though humans, of course, have been exploiting natural resources for thousands of years, the Industrial Revolution in the 19th century has produced an astonishing acceleration of the rate at which we are molding our environment. And industrial waste, urbanization, overexploitation of natural resources, habitat destructions are just a few examples of how we are impacting biodiversity. And actually, human activities have been suggested to produce even more concerning environmental change through global warming. And actually, global warming has attracted a lot of attention and is a very debated issue because it is supposed to potentially produce very catastrophic effects on human populations, because we know that even very minor temperature changes can have a large effect on crop production. And we also know that people that live in coastal areas may be forced to leave these areas if the sea level rises up. An additional source of concern related to this global warming has been that some diseases might actually spread outside the distribution, the current distribution area. And so either spread into new areas, or there might be some cases of emergence or re-emergence of infectious diseases. And this concern is particularly relevant for vector-borne diseases for obvious reasons. And these reasons are that these diseases are transmitted by mosquitoes or arthropods in general. And we know that these arthropods are very sensitive to temperature. So if temperatures goes up, you can speculate that perhaps these things might move to new areas and therefore spread the disease into these new areas. Another reason is that as temperatures goes up, these insects can complete more cycles within a given period of time, and of course this also increases the transmission risk. And so early work on human malaria has indeed suggested that this might be a real concern. There are a couple of examples here. So in this one, people have looked at the potential impact of climate change, global warming, on the number of months that are suitable for Plasmodium falciparum transmission. So this is the agent of human malaria. And they ended up with this clear pattern. So when they compared the current situation here to the predicted one, based on 3 different climatic scenarios, they ended up with the prediction that the number of months suitable for transmission should go up in the future. And another example here where they basically showed that transmission should be enhanced in Africa over most of the range. And they also suggested that actually malaria might spread into new areas in Europe and, well, overall in the Northern Hemisphere. However, these predictions have been challenged by the historical survey of endemicity of human malaria. So these are the real data. So these were the endemicity of malaria back in about 100 years ago, and this was the situation in 2007. And as you can see, contrary to these climatic predictions, the endemicity areas have been shrinking over the last century. So these are the change, and as you can see, there are very, very few spots, the red ones here and there, where endemicity has increased. So there is clearly a discrepancy between the models and the predictions that have been put forward by these climatic models and the actual data. And this discrepancy can be easily explained by the fact that humans have tried at least to prevent malaria transmission in most parts of the world. And even though many people are still paying a high toll to malaria, especially in Africa, it is undeniable that the infectious risk has really improved nowadays compared to 100 years ago. So I think that this example nicely illustrates the fact that human diseases, they're a difficult system to use when you want to forecast the effect of climate change on disease spread, because obviously there are lots of socio-economic factors that can play a confounding role. So I would try to convince you that vector-borne diseases of wildlife might be a more straightforward model system that can be used to assess the potential impact of global change. And they're very similar diseases, of course, so they share the same basic life cycle as human malaria. So this is the life cycle of the avian malaria. So the mosquitoes basically transmit the disease to disease-free birds, and then so once the bird is infected, the mosquito bites it again, and the cycle is completed. So very, very similar life cycle of compared to the Plasmodium falciparum. And actually, during the last couple of years, many people, a few different groups have looked at these avian malaria parasites as... Okay, so I was saying that in the last couple of years, a few different groups have looked at these avian malaria parasites as a suitable model to assess the impact of global change on the spread of disease. And so a couple of years ago, Laszlo Garazecki, he conducted an historical survey of worldwide prevalence of malaria in birds. So this is a quite huge data set. And contrary to the human data, he found that actually prevalence of infection tended to go up during the last decades. So just remember you that the situation for human malaria is exactly the opposite. And he also looked at the potential explanatory power or temperature anomaly, what is called temperature anomaly. And he found this weak but highly significant relationship. And here the different colors refer to the different continents, so Europe, Africa, North America, and so on. More recently, another study, Perez Rodriguez and co-workers, so they focused on one bird species, so the blackcap, and they sampled a bunch of different populations in Spain, and they looked at the three genera of avian malaria, so the Immoproteus, the Plasmodium leucocytosone, and then they looked at series of different environmental descriptors, so landscape, rainfall, temperature, geographical, so latitude, and they tried to see which one of these was the best descriptor of between population variability and prevalence. And as you can see, whatever the genus of parasite, temperature seems to really play the most dominant role. So temperature is really a major determinant of contemporary malaria infection, at least in these species. So we have also been doing quite similar work using another bird species, so the house sparrow, which is a very widespread and common species, and we focused on Plasmodium relictum, because this is by far the most dominant parasite species in house sparrows. And so we sampled a bunch of different populations in France, and again we looked at the predictive power of a few bioclimatic variables, And as you can see, we were quite successful because 83% of variation in prevalence was explained by these bioclimatic variables. And whatever the type of criteria that we used to score the relative importance of these variables, temperature turned out to be the most important one. And as you can see, especially mean diurnal temperature range was very tightly correlated with prevalence. So, okay, so if we use this information to predict the change in prevalence at 2 time points, so in 2050 and 2080, we end up with this map, and you can see that the model predicts that actually prevalence should go up in most parts of the country, with quite limited areas around the Mediterranean coast where the prevalence is predicted to go down. So relatively clear pattern here. So, but now the question is, and so what? So if we trust this model and we are confident that malaria is going to, avian malaria is going to spread during the next few decades, what are the consequences of this spread? Should we be concerned for the persistence of host populations or species? And I don't think this is so straightforward, because we know that animals, of course, do respond to an infectious insult, and an increase in parasite diversity, prevalence, or even intensity does not necessarily incur a fitness penalty for the host. And I think the best way to illustrate this idea is to look at the health by pathogen space. So what is this? Okay, so we start with one individual that carries a certain number of parasites, and this corresponds to a given fitness values. But another individual might carry more parasite copies, and therefore will pay a higher cost of infection and have a lower fitness. But another individual may have lower parasite copies and have a better fitness. So this line here represents the resistance gradient. So you have susceptible individual here, highly infected, poor fitness, and the resistant individuals here, low parasite infection and high fitness. However, you can also move along another dimension in this space, because you can have exactly the same intensity of the infection, but simply pay a different cost. And this gradient here is called tolerance. So basically you have different fitness payoff for a given parasite intensity. Another way of looking at tolerance is as a reaction norm of fitness across different levels of intensity of infection. So given that this is a reaction norm, this approach works with groups of individuals. So in this case, you have 3 groups of individuals, A, B, and C. This can be clones, genotypes, experimental groups, populations, or even species. And you can use the parameters of these regressional lines to describe these different things. So in this case, you can see that intercept is the same for the 3 groups. So this means that in the absence of infection, these 3 groups have the same fitness. You can also see that these 3 groups are equally resistant because the average parasitemia is the same. But of course, the tolerance is very different because the slope of these regression lines is very different. So C is by far the less tolerant group, where A is the more tolerant, because as far as you increase parasitemia, the decrease in fitness is very weak. Okay, so now the question is, at least that is interesting to me, is to know whether global warming can change the slope of the reaction norm between fitness and parasitemia. And I think that this is very likely because, of course, global warming does not only directly affect vector and parasite performance, but it does also indirectly affect other major aspects of the ecology of the interaction. And actually, when you look at the approximate causes of extinction due to climate change, it turns out that temperature is not the most important factor. So this year, Kale and co-workers reviewed all the known causes, approximate causes of extinctions. Actually, they looked at local extinction and population declines that were due to anthropogenic climate change. They also looked at natural climatic oscillations, but so let's focus on these two things, anthropogenic climate change. And so they looked at these approximate causes, for instance, extreme temperature, so desiccation stress, loss of aquatic habitat, and so on. And as you can see, the most important factor here is reduction in food. So the best predictor of the anthropogenic climate change impact on local extinction and population declines is through reduction of food. And of course, we also know that food is a major determinant of immunity, of infection risk and severity. There are plenty of examples involving humans and animal models. So I'm arguing that manipulating food quality and quantity actually offers an ecologically realistic and very simple way to explore the complex relationship between global change and disease severity. And so this is what we attempted to do with this, again, very simple experiment. So we had the host here, and we had two experimental groups. One group was fed with a supplemented diet that received protein and vitamins, and another group was fed with seeds only. And then these animals were experimentally infected with Plasmodium relictum. And of course, our hypothesis was that birds that were on a poor diet would have a weaker immune response, so they were less they should have been less able to keep control of the parasite infection, and indeed this is what we found. So this is the parasitemia, days post-infection for the two groups, and you can see that supplemented birds, so the rich diets, they were able to keep track of to control parasite multiplication much better because the peak was lower and occurred at earlier dates post-infection. However, when you look at the proxy of the cost of infection, surprisingly, it turns out that birds that were in the supplemented, so the rich diet, they suffered more than those that were in the seed-only diet. And this might be difficult to explain, but we believe that this result is due to the antagonistic effect that diet has on immunity. So what we believe is that animals that were in the rich diet, they were able to mount a stronger immune response, and of course this is good for you because you are more able to control parasite multiplication, but we know that immunity also incurs cost. And this idea is very clear when you look at, again, at the fitness by pathogen space. So you have the two slopes for the animals that were in the seeds-only diet and those in the supplemented diet. And there are two things that are clearly visible in these plots. The first one is that, again, birds that were in the seeds-only group were less resistant because the mean parasitemia was higher. However, they were much more tolerant to the infection because the slope is shallower than for the supplemented birds. So another question is, what is the best strategy? Would you prefer to be tolerant or resistant to the infection? So this is an open question. Something that is also quite important to take into account, I guess, if we want to have a relatively complex picture of what is going on out there in the nature, is that there might be also consequences for parasite evolution. And the reasons for this are very clear. So the prevailing view on the evolution of parasite virulence is that there is a trade-off between the parasite multiplication and the benefits that this parasite multiplication incurs in terms of enhanced transmission and the cost that is paid by the host. So over-exploitative parasite strains might have a benefit in terms of enhanced transmission, but if they kill the host, then the transmission stops, and of course the potential of transmission for the parasite is also zero. And of course, what we can speculate is that the tolerance breaks down the covariation between parasite multiplication and virulence, because tolerant hosts can withstand a much larger parasite population paying a smaller cost. So for these reasons, there have been a few attempts in the literature to propose or to suggest that tolerant hosts that pay a smaller cost of infection might be selecting actually for more exploitative parasite strains. And so we tested again this idea. So with another experiment, so we had birds that were split into the two groups. They were infected, and then we used the parasites that were grown on supplemented and seed-only birds to infect other animals that were again split into two groups. So here we have two factors that are fully crossed: the parasite environment, so the environment where the parasites were grown, and the host environment. And so these are the results. So black and red indicates parasite environment, the parasite origin. So those that were grown in seed-only birds, those who were grown in supplemented birds. And this is the current host environment. So those that were birds that were fed on seeds and were fed on supplemented diet. And as you can see, whatever the current environment of the host, parasites that were grown on seed-only birds reached higher parasitemia and were inducing higher damage to the host in terms of reduction in red blood cells. So actually, so again, this is independent of the host environment. So it seems that parasites that are growing on tolerant hosts actually might become more virulent than those that are in more resistant, those hosts that rely more on a resistant strategy. So I will leave it here with a few take-home messages. The first one is that perhaps vector-borne diseases of wildlife might provide an easier model to assess the potential role of environmental change compared to human diseases where, of course, these socio-economic factors may play a major confounding role. Something that I think is important to take into account is the potential for these environmental changes to shape the tolerance reaction norm. And the results are not that straightforward because we might end up with different pictures, probably depending on the parasite species, the host species, because these environmental changes may make hosts more or less susceptible to infectious diseases, more or less resistant or more or less tolerant, and these two things are not synonymous things. And finally, I think that in addition to exploring the direct effects of global warming on the spread of the disease, I think that further work should focus on the indirect consequences on host defense strategies and possibly on the long-term effect on parasite evolution. And I would like to acknowledge all my co-authors and the funding agencies and you for your attention.